It's my pleasure now to introduce our second speaker of the day, group leader, Dr. Lena Friberg. And we are moving a little bit further into the developing process of new antibacterials from what Oscar was talking very early on in the pipeline to clinical trials. And Lena is going to talk to us a bit about her work on analyzing the data from these clinical trials to have a more informative process for antibacterials for infections caused by multi-drug resistant bacteria in particular. Thank you so much, Lena, for being here with us, and the screen and floor is yours.
Thank you. I think, yeah. So is it, let's see.
Yeah, we see it.
Perfect. Thank you so much. And thank you for letting me come and speak here today. I'll just need to move all of you so I see the screen, or maybe it works anyway. So what I will do today is, as Eva introduced, is to talk about the other end of drug development and about the clinical trials for antibiotics and how we could potentially analyze such data in a better way so it would make drug development more attractive. So the work in this field here, it's Victor who has done what I will show today. So he was a PhD student admitted in 2017, so the first round of the UAC PhD students, and he's now doing an internship in Basel at Roche. So he has a little break, but will come back just in a month and finalize his PhD studies, which is anticipated to be in the end of the year. And I'm a professor of pharmacokinetics and pharmacodynamics at the Department of Pharmacy. So why is this important? Well, you probably have come across and heard before that it's not just to get a compound that seems to work in vitro or in vivo, it also needs to have properties that it is efficacious also in clinical trials. And there are many things that can happen during the course of clinical trials. As you know, they are very expensive. And we need to find some ways to reduce the cost so that it becomes more attractive. So you may have seen graphs or other sketches similar to this, that there's a lot of compounds that may not reach the clinical development, but it does actually look much better now compared to what it did some years ago. So a big problem is the gram-negative bacteria, and that's what we need to find new solutions for, or new compounds that work against this class. And there are still relatively few antibiotics in the pipeline, so we need to find some ways to make it more attractive. And one important aspect is to reduce the need for clinical data, or how much clinical data that we really need to make this happen. And then we may need to rely more on animal studies and also studies in vitro systems. So that's what we in general try to do. And this is a sort of schematic that John Rex outlined quite a few years ago now, but the idea was here that we don't need these two phase 3 trials. So phase 3 trials are the really big trials where you compare to another compound or treatment which is already available to patients and try to prove them that you are at least as good as those. And these require quite a few, several hundred patients each. So it took a very long time to accomplish these trials, and of course it becomes very expensive. So what has actually happened is that it's more towards this tier B here now, where it's maybe sufficient with one large phase 3 trial, and then you can combine that with several smaller studies. But the idea is that maybe the more preclinical data we have, or the more informative preclinical data we have, we may be able to reduce the clinical information and rely on other aspects. And the animal rule here is like for compounds against anthrax, for example, where you can't really study at all in humans. But we probably, for most infections we are talking about, we will still need some efficacy data in patients also in the future. We're trying to move on this end. And our approach is to build mathematical functions that can describe the relationships between the animal studies and the clinical studies. So we focus on the pharmacokinetics and pharmacodynamics, and that's really how the dose regimen, so the dose amount and how often you give the dose, how that is related to the concentration in the body, and also the concentration that actually reaches the sites where the bacteria are, and then how that concentration is related to the effect. And we are really interested in the time courses of drug effects. So how does it look over time? And it's quite complex in the way that the bacteria multiply quickly, and then they also can be killed off quickly. So it's a really dynamic systems that we try to understand here. And therefore we need to, in general, describe this by models. We can't just measure one time point and think that we know how it looks like. So once we have these models, we can also start to explore different scenarios. So what if we give another dose? What happens then? How will the effect then look over time if we change something in the system? But then we have the problem for these type of bacteria that we are interested in, that it's generally difficult to measure them in humans. So that's why we've focused a lot on the in vitro and animal studies before. So how can we actually get good data from patients? Because if we look into what type of information we get from clinical trial studies, then in, for example, the gram-negative space, it's generally very low information data. So the primary endpoints are typically dichotomous. It's like yes or no. So from one patient, you get one sort of data point, yes or no. So was the patient cured, or did it fail? The treatment failed. Or it could also be, is the patient dead or alive at a certain day? So you can understand that that also increased the number of patients we may need, because we just have one sort of information data point from one individual. And so one way to do it slightly more informative would be to treat this data as time to event data. So how long does the patient survive? If you have these serious infections, that may be important. Or yeah, when can the patient be discharged from the ICU, for example, or from the hospital? But even more informative would be to use longitudinal measurements. So we look more into the patient's general disease status. And here we have SOFA score, which I will come back to, and also the PASH score. You can also think of fever, but of course it's difficult because you get medication towards the fever, and that may not be so reliable from that perspective. So how can our pharmacokinetic and pharmacodynamic models be of value? So we can predict the outcomes from preclinical data. That's the ideal case, and that's what we are trying to do, and find these relationships that work well. And with our techniques, we can use the time courses rather than just this fixed time point, and also account for that the time course in a test tube may not look like a time course in a human. So the idea is to sort of go through this chain to make better predictions, but what and how to link to these patient outcomes that are used in clinical trials, that's sort of a challenge that we try to start to investigate, I would say, in this project that Victor is working on. So we want to improve the study designs, and what we mean with the study designs is a dose and dosing interval, the study duration, how many patients, when should we measure different things. And the main focus of Victor's work is really see how can we analyze data from these type of trials. So hopefully come up with some suggestions that could be more efficient. So for this work, we had data from a clinical trial, the AIDA trial. And the main focus here was really to look into if a patient was infected with the carbapenem resistant bacteria and then would be treated with colistin. Would it be a benefit to actually add also meropenem to the treatment? Because what has been shown in vitro was that the colistin may open the membrane and the meropenem can get in and kill the bacteria. So there are synergistic effects of this combination. And we also did a systematic review to look into the previous in vitro results that have been published. And it was clear that especially for Acinetobacter baumannii, there was a synergy rate. So this seemed very promising then to do this combination in clinical trials. But there was still some doubt about this. And we set up this trial, and it was 406 patients that were recruited during 3 years, and this was at 6 different centers. So it was in Israel, in Italy, and also in Greece. And the requirement for these patients to be included in the study was that they should be resistant or have bacteria, be infected with bacteria that were resistant to meropenem, so an MIC above 2, but be susceptible to colistin, so an MIC less than 2. And then results came after these years and a lot of efforts. And there was actually no statistically significant difference between the 2 study arms. So there didn't seem to be a clinical benefit of adding meropenem to the colistin treatment.
Lena, I have a question here, if you don't mind.
Sure.
Do you think that it's a numbers thing? Do you think if more people have been recruited, maybe will have affected the results?
Possibly, or eventually. I think there could have been, because there was some numerical difference, but I think the difference would probably not be clinically meaningful. So if you just look at the numbers, there seemed to be a small difference. But then, of course, if you would have twice as many subjects, then maybe it would have been clinically significant. But then the difference would still be so small, because what the clinicians also worry about is that giving meropenem when it's not needed may actually trigger even more resistance.
I understand. Thank you.
So what we then, as interested in the PKPD, wanted to find out was if the colistin concentration may be important. So it could be that those that have a low colistin concentration, they would have a more poor outcome. Well, that would be sort of the general idea in PKPD, that the higher the concentration, the better the effect would be. So we had measured colistin concentrations from each individual, and as illustrated here, this is just predicted out how it would look in all the different subjects over time, the colistin concentration, and you can see that it varies quite a lot between different patients here. So some has very low and definitely lower than the MIC, and some above. So it's in a quite wide range. So we then did a time to event analysis to look into what are the predictors of survival. And the most important one, that was this SOFA score, that is then measuring the overall status, disease status of the patient, you can say. So it's score of organ failure or how well you are performing. And again, I will come back into more detail about that later. So that was the most important one. But then even if we have accounted for that, it's the death increased with age, as you would expect. And then also, if the patient was infected with Acinetobacter or Pseudomonas, there was a worse outcome than from other types of bacteria. But what we were really interested in was, of course, to see here if the concentration made a difference. And then it seemed that actually there was not much difference here, or the other way around even, so that the higher the colistin exposure, the lower was the survival, so it's actually a higher risk of death. So this was maybe a bit surprising, but at the same time we should remember that colistin or its prodrug is eliminated by the kidney. And if you have a poor status, your kidney function is reduced, and therefore that triggers the colistin to go up. So it's a quite tricky situation here to sort out what is the relationship to what. But anyhow, we are very interested in the kidney function and so on, because that's what really is dose limiting for giving colistin. So we want to find this sort of sweet spot where we have a plasma concentration which is killing the bacteria but not causing toxicity to the kidneys. So for this, Victor has developed a model describing the serum creatinine in blood over time, because that is a measure of the kidney function, or it's related to the kidney function. So if we have a reduced kidney function, then the serum creatinine levels increase. So we had that type of model, and with this we can then predict out what is the suitable or target concentration, sort of, or an upper limit of the concentration where too many subjects will have the acute kidney injury, which we don't want the patients to have, of course. And we can also see here, this is just predictions from subjects within the trial if they had received different concentrations. So you can see here the range of concentrations. So the higher the concentration, the higher is the probability of having acute kidney injury. So with the help and feedback from clinicians, we think that with this we will be able to have some threshold, and then that can guide us in what should be the upper limit, and also for how long should the patient be treated at maximum, because you can see that the risk is increasing during the treatment period. So now returning to this SOFA score that I have touched upon before, it's something that could provide longitudinal information, so information over time in a patient about the disease status. And the scale this score has 6 different items. So it's 1 for each organ system. And then each organ system has, you score by, it's 5 different scores, so from 0 to 4. So the total score for SOFA then go from 0 and up to 24, where 24 is the worst, and 0, then you're fully healthy. And this is a score with integer values. So you can't have a score of 5.3. It's 1, 2, 3, 4, and so on. So this is a little bit of a challenge, or a different way of looking at it as when you come to building a model. And also we wanted to explore to see if there are some of these subscores, or some of these items related to certain organs that are more informative than others. And we could identify a few organs that had more information than others. But on the other hand, when we tried to use that information and optimize the score and weigh in different ways, we couldn't see that actually was a difference or an improvement. And therefore, we think that the SOFA score is probably quite good as it is. And this more cumbersome way of looking at it, which is called item response modeling, may not be needed for this type of data. So instead, we could go and analyze the data in another way, which is called then the bounded integer model. So here we can also model the score over time and look into the mean and the variance and how that is changing depending on different patient factors. So that's something that Victor is also sort of compiling now, and the last pieces of them, which will be presented. And the last piece of his work is related to something that we call multi-state modeling. And this is a statistical method where you can look into different states. So the patients would, for example, start here with a positive culture. So they have the bacteria, and then as time goes by, they may turn negative. So there are no longer bacteria. And if it's worst case, then of course they will die. But they can also die, of course, if they also have a negative culture. But with this type of structure and model, we can sort out better where is the drug effect and what is causing what. So this is a way of accounting for competing risks, and therefore maybe a better and more informative way of describing the data. So I'll also mention very briefly another project which is not related to Victor's work, but it's connected to the AIDA study, because what is unique there is that we have a lot of individual information. So we have, as I mentioned earlier, the plasma concentration time profiles from each patient, but also we have information about the infecting bacteria. So in this work, we focused on the Acinetobacters because they were most patients had Acinetobacter infections. And each of this bacteria that were infecting then different patients, we had the MICs and also synergy measured. So we could look into that as well. And also the fitness of the bacteria has been tested in a mouse model. So we know how well they grow in vivo. So all this information has then been included in an earlier developed PKPD model describing the growth and the killing of the bacteria. And by making use of all this individual information, we can sort of see how the bacteria may grow or decline over time for each individual. And we took the last observation here, we're at the 24-hour value, and then correlated that to the probability of clinical failure. So again, there are other factors, of course, that are related to if the treatment was successful or not. Age, SOFA score, and here the Charlson score as well. But then we could also see that for those where these factors, the combined factors, if we had a bacteria that were less than 10 to the power of 8, then we could see that the failure rate would be lower compared to if the patient had a higher or very high load after 24 hours. So these are numbers, you can't really say that these are the numbers in the patients, but so this is more on a relative scale. But it's quite interesting and quite unique data set, I would say, that you have all this information within a patient. So to summarize, we think here that Victor's work will provide a sort of a toolbox for analyzing clinical data of antibiotics against AMR. So we have looked into the possibilities of using time to event modeling for modeling time to death, and also predict acute kidney injury. And the SOFA scores, we have looked into how that could be best characterized, how should we analyze such data, and found that the bounded integer model seems to be a promising method. And once you have set that up, it's a quite straightforward model to use. That seems suitable and promising. And then we will also explore more the multi-state model. That is also likely a more informative way than just looking at failure or not. And then taking into account the PKPD data and also the information from the individual patient and the bacteria that they actually were infected with, we think that we can provide more information on the clinical outcome. So ultimately, we hope that this could lead to fewer trials and fewer individuals per trial especially, and that this would lead to a reduced cost and reach the patients ideally faster and also the market. So that was what I planned to present today, and I didn't provide a full slide of acknowledgement here. I focused on the work that Victor has done and the supports that we have received for this work. And now I'm happy to take any questions.
Great. Thank you so much, Lena, for this presentation and seeing the development of Victor's work from the very beginning to now. Let's see if anybody either wants to open their mic and ask a question or write it down. Meanwhile, maybe I can ask you a question that kind of came to mind while seeing your presentation. Is you are trying to come up with models that can give more information and therefore reduce the amount of clinical trials or the amount of people. But as I understand, the clinical trial process is something heavily regulated, and the results of the clinical trials is what then the regulatory agencies uses to either approve or not approve or allow a new treatment or not. How do you see these changes that come from research, the research that you're doing, into being integrated into the system and accepted by these regulatory agencies? Also because they are different ones, right? It's not the same thing as bringing a drug to the FDA and the US market to European EMA market. How do you see this changing in the future?
I think it's important to have a really good communication with them. I think like this figure that I showed in the beginning with the tier A and B. So I think it has already been a switch from A to B. So I think that's quite accepted. And they also acknowledge, if you look into guidelines, that the preclinical data is of importance. Then it's just to get that more precise and reliable. I think we need to show with examples that it works better, but I think they are also eager to get more information. I think that there is also, of course, the safety side. So you need to study the drug maybe in a larger population, but that could be for a more other type of infection, which may be more common because it's still difficult to find these patients that has the difficult bugs. Mm. Maybe if it works also with the more susceptible bugs, then we can study a larger population and then get the safety part from them. So
yeah, what I understood talking with John Rex in particular is that that's the issue, that sometimes if you do it in a sensitive bug, like you already have a drug that already works for that. So you're basically getting either the same result or not superiority result. So it's the same. So then it kind of doesn't pass to the next stages, which would be to use it in patients that really have a resistant infection, because the pool is so much smaller than what you will find for a sensitive or bug, right? So it's kind of hard to go through all those stages in that regard. there's not yet any other questions, but I have another question. So you were showing the data on patients that are treated with colistin and how the concentration changes. I don't know if I understood correctly, that concentration is measured in time in each patient in the serum so that you get the particular concentration of the drug. And as I understood, the differences in the concentration could be because of the clearance of each patient on the drug, but are these patients, all of them, given the same amount to start with?
Yeah, so in that study, they had exactly the same dosages, except for those who had a poor kidney function. Then there was a reduced formula for how to reduce the dose. And we could see that it did sort of lower the exposure compared to what it would have been if they were given the full dose, but it probably should have been even lower because they still had a somewhat higher concentration, those with the poor kidney function compared to those with a more normal kidney function. But then in this disease area, it's also so that many patients, when they are this sick, they actually have an increased clearance of the drug, especially if it's younger patients. So it's like the body opens up to fluid is sort of moving around and also then empties quicker. So the clearance of the drug is often then also higher, and then the colistin concentrations even lower. So they may actually need a higher dose if we think that if it is a concentration effect relationship there. But again, that's also a population that may be more affected because the body reacts in that way. So it's both those with a poor kidney function and those with a normally high kidney function that may be the most critical patients.
So do you see in the future that the colistin treatment could be really patient to patient? It will vary in the concentration that they're given.
You mean, in the future? Or
yeah, on the future, like if we could move to a situation where every patient will get the exact amount of drug that it will work best for them, given their whole overall health or their status, if they're clearing more or less or something like that.
I think in the ideal world, we would try to have an effective concentration that is killing the bacteria, still not causing too much kidney toxicity. But it's the difficulty is to identify that dose. So ideally you would have patient factors, so know what kidney function they have and what weight and so on that may go into the equation, and then you will be there. But what we have seen, it's quite a high variability also, and I think especially it's also with the prodrug, the CMS, so colistin is given with the prodrug, which causes even more variability. So then you may need to take a sample and having that assayed, and then you could adjust the dose. But given that the patients quickly change a lot also in the kidney function, I think it's a really tricky part. So if there would be some other drugs better than colistin, I think that could be.
Yeah, I was gonna ask, is the pattern of having a lot of variability in the concentration of the drug happens also in other drugs, in other antibiotics?
Yes, but I would say that maybe for comparing to those other antibiotics I worked with, I think maybe colistin is more unpredictable, that it's more difficult. Otherwise you have often a very high dependence on the kidney function. But I should say that colistin is still a lot used. Even these results, we were thinking that maybe it's not so much effect, but it still seems to be what is used, for example, in Israel, where they have a big problem with Acinetobacter being resistant to carbapenems and so on.
I guess it's because there's no other option for them to use, right?
Exactly. Yeah.
Great. Thank you so much. Does anybody has any more questions for Lena? I think I've spoken enough. It doesn't seem like there are any questions, but obviously you are reachable by email. I guess you can find Lena's email in our website as well and learn a little bit more about the project that Victor was doing in case you want to review that. But I think if nobody else has anything to bring up, then I think we're gonna close today's webinar. And I thank you, Lena and Oscar, so much for presenting your work and bring to us a bit more of the research that we're doing at the center. Thank you so much, everybody, and I'll see you back in 4th of March will be our next webinar. And meantime, you can enjoy the podcast as well. Thank you so much, everybody, and bye.